Welcome to FlyOde, an ode to fly development. FlyOde is a platform for researchers of the developmental biology of Drosophila. It provides an overview by displaying the gene and protein interactions during the development of fly organs. It gives deeper insight into the mechanisms at work through filtering the network according to the expression pattern of a specific gene at a specific developmental stage. We encourage you to contribute your knowledge to ultimately have the collective knowledge about fly development organized in one network. Find out more on how you profit from this in the About section and how you can contribute later in this video. But let's first have a look at the network to get an overview. Click Go to Network and wait until the network finished loading. In the network display area, you see all genes or proteins as nodes and their interactions or edges as lines with arrowheads for positive or activating interactions, or T's indicating negative or inhibiting interactions. Undirected edges indicate protein-protein complexes and do not have a shape at their ends. See legend for color coding of genes. The network currently represents a fraction of all interactions that take place during development of various Drosophila organs, at this stage mostly the Drosophila melanogaster retina. The network layout is arranged to indicate hierarchy from top to bottom and developmental progress from left to right, such that genes and proteins expressed at early stages, e.g. the larva, are located at the left, and those expressed at later stages, like pupa or adult, towards the right. Thus, the nodes on the bottom right are the rhodopsins, which are expressed towards the end of retinal differentiation. Single nodes or groups can be selected and moved. On the bottom right of the network area, tools can be selected for zooming in and out and for panning. As you hover over a node, you see additional information in the info area below the network area. The gene symbol, name and alternative names, the flybase link for more information, and the expression pattern, which is manually extracted from the literature, is shown in its native annotation syntax, which is relatively intuitive after a short-term usage period. Click on Expression Pattern to bring up a table explaining the annotation terms. It is given by the developmental stage, larval stages over pupa to adult in combination with cell types, or in relation to the morphogenetic furrow. When given in the annotation, the phenotypes of mutants or overexpressors are shown. The gene ontology annotation informs about the biological process in which the gene is involved, in which subcellular component it is located, and which molecular function it has. Finally, the references are given. As you hover over an edge, in the info area the formal annotation of the interaction is displayed along with its reference. Now let's see how to get information about your gene in the network. Let's first reload the page to restore the initial layout. Click on the rightmost area of the screen to open the search and filter area. Locate the search box and make sure that the radio button gene protein name is active, which is the default state. Start typing your gene protein name. As an example, we choose spineless. After typing 2 letters, a list with available hits is displayed. Continue typing to narrow down the results. Use the arrow keys or the mouse to select your gene. Click on the double arrow to display the network area. Spineless is highlighted. It is located on the right part of the network, indicating its expression during pupal stages. This is confirmed by checking the expression pattern entry in the info area, which shows its expression in intermediate and late pupa yellow R7 photoreceptors. Let's rearrange the network and have a closer look at its interactions. Spineless directly activates rhodopsin 4 expression, and in a complex with Tango activates defective proventriculus to inhibit rhodopsin 3, rhodopsin 5, and rhodopsin 6 expression in yellow R7 cells. Furthermore, it also affects rhodopsin expression in R8 cells by an indirect inhibition of melted. Now I will show you how to use FlyOde to represent the differentiation of a specific cell type. More precisely, we will filter the network to only show nodes with a specific expression pattern and highlight a subset of them. In this example, we are interested in the development of the inner photoreceptors. Go to the Search filter area. Select photoreceptor cell from the pull-down menu Cell Type. Select at least one photoreceptor cell from the next pull-down menu Any photoreceptor cell. Press the Filter button. Display the network area. Genes, proteins that are expressed in neither R7 nor R8 are removed. The current network contains nodes that are generally expressed in photoreceptors, e.g. ILS. To make the network more specific for inner photoreceptors, we add another filter by clicking on the plus at the end of the filter line and connect the two filters with the Boolean operator NOT. Select photoreceptor cell and at least one outer photoreceptor. Which in combination with NOT means none of the outer photoreceptor cells. Press the filter button. Especially nodes from the left part of the network were removed, reflecting the expression of more general photoreceptor differentiation genes during larval development. Of the remaining nodes, which ones are specifically expressed in yellow R7 cells during pupil development? Add another filter line using AND. Select pupa from the Stage pull-down menu. Photoreceptor cell, photoreceptor cell R7, and yellow from all subtypes. Then press Select. Display the network area, so those nodes from the previously generated network are highlighted that are expressed in yellow R7 cells. Now imagine that we want to perform an immunostaining and need a marker for a specific cell type, say R8, in the pupil stage. How could you use FlyOde to figure out which antibody to use? I hope I could inspire you to use FlyOde to answer your own questions and to plan projects. Finally, I would like to encourage you to contribute by submitting a notation about your own favorite genes via our online form. You can find it in the top left corner of the network area. It is easy to fill out and essential for the usefulness of the platform. Enjoy, your FlyOde team.